Welcome back. We are looking at a situation where there is a simultaneous concentration gradient and electrical potential gradient. We had derived the Nernst equation yesterday, as well as we saw the Nernst-Planck equation. Essentially, we looked at the condition of electrodiffusive equilibrium when there is no net movement of an ion under the combined influence of an electrical field as well as a concentration gradient, an electrical potential gradient as well as a concentration gradient. Okay, that's what we had seen in the previous class. Let us continue further. In this class, we are going to look at mobility of ions across a membrane. Okay. This is highly relevant, extremely relevant for the biological engineers and of course in the human body, as we will see. We saw earlier charges and their dynamics, or at least we stated earlier, charges and their dynamics are responsible for our ability to sense our environment through sight, smell, taste, touch, hearing, the five senses, and so on and so forth. Okay. We said that all these five senses work by the charges and their dynamics. The dynamics of charges are essential for the functioning of our nervous system, our brain, our heart. This is also we saw the same sentences. More specifically, our senses work because of the mobility of ions across neural cell membranes. Okay, neural cells, neurons, they're large cells, and our senses work because of the mobility of ions across the neural cell membranes. Some proteins in the membrane are passive, they do not require energy for transport, such as ion channels, or active, which require energy, such as transporters for the transport of charged ions across the membrane. In other words, there are two ways by which ions can get across the membrane. Apart from regular diffusion, the rates of ion diffusion through a lipid bilayer membrane are very, very slow. Okay, especially charged, and there is a hydrophilic head, a hydrophobic core, and a charged particle moving through such an environment is very difficult from the point of view of the nature of the molecules involved, and therefore the permeability of ions across a lipid bilayer membrane is very, very small. Okay. So low that we usually can neglect it unless you are considering very long times and so on. So, in the absence of reasonable transport rates of the ions across the lipid bilayer of the membrane, it is through the proteins that the ions can get in and out of the cell. There are two ways by which that happens. One is a passive way through the ion channels, okay, and an active way through the transporters. Passive means it does not require energy to transport an ion from one side to the other. Active, it requires energy. It requires the breakdown of ATP to provide the energy for transport of ions across that protein. Okay. Both are involved. Due to their very nature, the charged species cannot permeate through the nonpolar lipid bilayer core at reasonable rates, although they can permeate at low rates, very low rates. What we discussed in detail just now. During ion movement across the biological membrane, or the charge flux across a membrane, recall charge flux across a mass flux across a membrane many chapters ago, at least two driving forces are present. One driving force we've already seen. Here we are bringing in another driving force. These two driving forces are the potential difference. If there is a difference in charges across a distance, in this case a membrane, there's going to be an electrical potential gradient, right? There's a potential difference across a distance, there's a potential gradient there. And the concentration difference of the ion under concentration. Okay. Both these are going to contribute. The presence of the membrane, it's essentially a medium, does not interfere with the manifestation of charge effects. This we already know. Okay, it's a field effect. So, and as long as it doesn't effectively shield the field and so on and so forth, it's not going to play a role in the manifestation of the charge effects, the presence of the medium, in this case a membrane. Thus, the Nernst equation that is valid for ions in solution at electrodiffusive equilibrium must be valid for the distribution of ions across a membrane. Nothing prevents it, right? Think about it. You have a medium, liquid medium, let us say, in which we derived the Nernst equation. Here, we're going to replace that liquid medium with a membrane. That's it. Let us consider only one ion, say potassium, first. Okay. Let us develop our understanding with one ion, and then we can extend that to the relevant ions. There are not many, by the way. The Nernst equation gives, just direct application of the Nernst equation, the potential in the intracellular space minus the potential in the extracellular space is nothing but minus RT by ZnF ln of the natural log of the concentration in the intracellular space divided by the concentration in the extracellular space. Okay, this is what the Nernst equation tells us. Intracellular, extracellular, of course. This number is 6.1.2-1. Okay, let me again repeat that this numbering may not be continuous in these lectures, but these numbers refer to the numbers of equations in the textbook, and to provide you with easy reference to textbook material, I am using the same numbers here. That I suppose would be helpful. This, we said, is valid at electrodiffusive equilibrium, and therefore this equation describes the equilibrium condition when there is no net transport of potassium ions. That is, when the potential across the membrane is able to balance the driving force due to the concentration difference in potassium between the inside and outside of the cell. Okay. So, see, there are two things. One is there is a difference in potential across the membrane. Two, there is a difference in concentration of potassium across the membrane. Both are going to drive. Okay. Opposite ways, the same way, whatever it is. And when there is no net transport of potassium ions, it means these two are balanced out. The sum of those equals 0. That's what we've seen. Okay. I'm just re-emphasizing that. The potassium ions that traverse the membrane are usually held on the other side of the membrane. They don't go into solution due to the charged head groups on both extremities of the membrane cross section that attract ions to reside on the surface of the membrane. This is just for some detail. We are not going to get too much into this particular aspect of it being on the surface and then going into the extracellular space and so on and so forth. But you need to at least know that this exists. And so a potential difference is set up across the membrane due to the difference in the charges on both sides of the membrane. This is known as the membrane potential. The potential that is created due to the charge difference across a membrane is called the membrane potential. The video, the neuronal membrane ion flow and concentration gradient. This nicely explains whatever I have said here. This will be a nice reinforcement for the understanding of this principle. Let's work out an example. Okay, we'll take, in this case, I've given you some information because it is new, and then I'm coming up with an example, just a variation of the theme. A certain cell was found to have the following equilibrium concentrations of potassium under the conditions of the experiment conducted at 37 degrees C. The intracellular concentration was 120 millimolar, the extracellular concentration was 2.5 millimolar. This is very standard for neural cells, the human body. So these could be taken as thumb rule values. There will be variations, of course, but around this you have an idea that in the absence of any other data, you could assume these. Assuming that the membrane is permeable only to potassium, estimate the equilibrium membrane potential. Okay. This is the problem. Pause the video for a few minutes and think about the problem. and then we'll take a look at the solution. Pause, please. Okay. Hopefully you have a better mental picture of the situation here. A certain cell was found to have the following equilibrium concentrations of potassium under the conditions of the experiment. Intercellular concentration, extracellular concentration given. You are asked to find the equilibrium membrane potential, assuming that the membrane is permeable only to potassium. So what is needed is an estimate of the equilibrium membrane potential. And what is given or known are the intracellular and extracellular concentrations of potassium at equilibrium. So how do you connect the known to the needed? We know from equation 6.1.2-1, our Nernst equation, that is. It relates to two, right? It relates the equilibrium membrane potential to the concentrations. So this is the equation that relates that. So as long as we find all the terms here, we would have whatever we need. The left-hand side is the membrane potential. Potassium is the only ion in the concentration, and therefore the Nernst potential of potassium is the membrane potential. Okay. In the rare case, we are looking only at one ion to understand it better. So Nernst potential of potassium equals the membrane potential in this case. This is the Nernst potential, by the way. And if we substitute the given values, we get intercellular potential, electrical potential minus extracellular potential. R is 8.314. T is should be in Kelvin, so 37 degrees C, 310 Kelvin. Zn, potassium has a valency of 1, a charge of 1, so that is 1. F is 96,487. This we have already seen in the previous chapter. Ln of intercellular concentration of potassium is 120 at equilibrium. Extracellular concentration of potassium is 2.5. Both are millimolar, so they will cancel with each other. We don't have to change the units here. 120 by 2.5. So this will turn out to be, if you crank the number out, minus 0.1034 volts, which is nothing but minus 103.4 millivolts. Okay. So this is a typical value, not a typical value, but if you consider one ion, it is a typical value, especially potassium. Now that we understand the distribution of a single ion across a membrane, let us consider the other ions also. Do we have time? Yeah, we do. And I don't think we are too tired. So let us consider the other ions also. The ions that are usually considered important are potassium, sodium, maybe calcium, chloride under extreme cases. Okay. There are reasons for each one of these. In fact, most of the initial development in this field of neurophysiology happened only with potassium and sodium. Okay. And then calcium came in under certain circumstances. Chloride is rarely used, and beyond this, nothing else is used. Normally speaking, an undergraduate need not even go beyond these. Okay. So the initial developments in the first of the 20th century considered only potassium and sodium ions, and the results predicted by the models were close to the experimental values for the type of cells that were studied. That's the reason. This is good enough a model. The chloride ion can passively pass through the cell membrane through its ion channel, of course, and usually the membrane potential is close to the Nernst potential of chloride. When it is close to the Nernst potential of chloride, the movement of chloride ions, then there's not going to be no net movement of chloride ions. That's the reason why chloride is not considered too important. It's considered at times, but not usually. The chloride ion current is very small and thus can be usually considered to be comparatively negligible. Also note that we are considering only passive transport of ions across the membrane. These models that we are looking at or developing they've already been developed, we are covering in this course, considering in this course, discussing in this course. These are all for passive transport of ions. That is, transport with no expenditure of energy. The ions that cross the membrane through active transport, such as the hydrogen ion, are not relevant for passive transport models. They cannot be explained by passive transport models because just not in the scope of things itself. We will not consider the active transport of ions across the membrane in this course. Of course, it is very important. Okay. Only thing is that for our purpose, which is transport and so on and so forth, we are not going to get into the complications of the active transport. Okay. So, if needed, of course, there's a whole body, huge body of information on this. You can go and read up. We will not consider active transport. We will limit ourselves to passive transport. Now, let us consider multiple ions, and when the equilibrium occurs, we look at it a certain way with certain assumptions which turn out to be valid, and such an equilibrium is called Donen equilibrium after the person who is known for it. Okay. So, this is a typical situation with multiple ions, or at least sodium, potassium, calcium. Multiple ion types contribute to charges on either side of the membrane. We all know this. In other words, the membrane potential depends on the dynamics of multiple ions. When only one ion was considered, that is potassium, the membrane potential was the potassium Nernst potential. We saw that when there was electrodiffusive equilibrium of potassium ions. So, that itself was good enough for us to predict, and that we said that's only for an understanding. When there are multiple ions, which is usually the case, closer to it is reality, the electrodiffusive equilibrium or no net movement of all ions. They could be individual ions moving. Only thing is that if you take the sum of ions moving to this side, or the rate at which ions move to this side, equals the rate at which ions move to this side, both in terms of charge and the numbers and so on. Okay. So, all put together, it's essentially charge movement that we are looking at. So, at the electrodiffusive equilibrium or no net movement of all ions would occur at a certain membrane potential called the resting potential, which may not correspond to the Nernst potential of individual ions. Okay. So, Nernst potential means that for that particular ion, the forces cancel out. Okay. The forces cancel out, so there is no net moment. Whereas now there are multiple ions that are contributing to the membrane potential. So, that may not equal the Nernst potential of any one ion. So, there at some potential, membrane potential, there is going to be no net movement of any ion across the surface. There will be individual movements, but there is no net movement. Hodgkin and Huxley in 1952, for a totally different purpose, they assumed that the currents of each ion are independent of the other ions, and this assumption worked very well. Okay. So, this was assumed that the currents of each ion are independent of the other ions, which means the current of sodium doesn't interfere with the current of potassium, that in turn doesn't interfere with the current of calcium, that doesn't interfere with the current of chloride, is what they assumed. Okay. There is no strong basis for this. It was an assumption, but that assumption was turned out to be a very good assumption. Okay. So, with this assumption, you could get a nice handle on the treatment or the understanding of this. Using this assumption, we can use the Nernst equation to relate the intracellular and extracellular concentrations of each ion separately when there is no net movement of ions at the resting potential. Okay. Because each ion is independent, the Nernst potential for the Nernst equation for each ion must be valid separately. So, that would take care of those things. Let us go further, then the significance of this will become clearer. The intracellular and extracellular concentrations of each ion at resting potential will be different from the values obtained when only one of that ion or when only that ion is considered for transport across the membrane. Okay. Let me read this again. The intracellular and extracellular concentrations of each ion at resting potential will of course be different from the values obtained when only that ion is considered to transport or move across the membrane. Okay. And Hodgkin and Huxley assumption works probably because the ion currents that arise due to non-equilibrium situations under resting potential conditions and consequent charge loss or gain from or by the cell is reasonably small for the time scales normally associated with the cell dynamics that I've studied. I'd leave you this sentence with you. When you get to a certain comfort level and understanding, you can go back to this sentence and try to understand why this could be happening. Okay. You are just about getting comfortable, so I don't want to confuse you, load you further with this also. So I'm not going to take too much time to explain the reason why this works. It works. Okay. In other words, we can assume that the resting potential, which is V intracellular minus V extracellular, equals minus RT by ZKF ln of CK intracellular divided by CK extracellular, which again equals minus RT by the valency of sodium times F ln of concentration of sodium in the intracellular space divided by the concentration of sodium ions in the extracellular space. Because each ion is independent, we can write the Nernst potential for each ion separately. But whatever is intracellular minus extracellular voltage is the same situation. Whatever you consider, that is for that condition, we are writing these equations. So the left hand side would be the same, which again equals minus RT by ZCAF ln of intracellular calcium concentration divided by the extracellular calcium concentration. Therefore, we could equate all these. Left hand sides are the same, therefore we could equate all these. If you do that, if you're going to equate this, then A ln B is ln of B power A, and therefore this goes to the index here, it gets raised to the power here, raised to the power here, raised to the power here, and then the arguments can be equated because they're all natural logs. If you equate the arguments, CK intracellular divided by CK extracellular raised to the power of 1 by ZK, RT by F is going to cancel throughout, equals intracellular sodium concentration divided by the extracellular sodium concentration raised to the power of 1 by Z, the charge of Na, equals the intracellular calcium concentration divided by the extracellular calcium concentration raised to the power of 1 by ZCA. Okay, this is how we got it. This, if you put in the numbers, the charge on potassium is +1, the charge on sodium is +1, the charge on calcium is +2, right? And if you take chloride, it will be a -1. Okay, so the sign goes along with it. CK intracellular divided by CK extracellular equals CNA intracellular divided by CNA extracellular equals the intracellular calcium concentration divided by the extracellular calcium concentration raised to the power half. Equation 6.1.2-2. Okay, and this is the condition for Donen equilibrium, which arose out of the application of the approximation by Hodgkin and Huxley that the fluxes of ions are independent of each other across a membrane under conditions of equilibrium. Okay, I think we will stop here in this class where we looked at Donen equilibrium and continue in the next class. See you.